Right, I'd like to welcome everybody to this MIDAS seminar today. As we begin to think about spring, we're delighted to have with us Dr. Anne Plant, who's a fellow at the National Institute for Standards and Technology. She was previously chief of the Biosystems and Biomaterials Division there. She's also been at the White House Office of Science and Technology Policy, which is the thing that sets the national agenda for science and technology. Dr. Plant works on cell imaging and understanding biological response in cells. She is an expert in metrology and how one thinks about measurements, and therefore, in terms of how we should be reporting data in scientific experiments. And we're really excited to hear what she's going to tell us about this. Dr. Plant is a fellow of the American Association for Advancement of Science and of the medical and biological engineering. Before I turn it over to her, just want to tell people that we have Vipin Kumar, professor from the University of Minnesota, next week. Vicky Bogan from Cornell the week after, and Ben Wellington from 2 Sigma on April 19th, talking about data visualization. Want to thank our sponsors and affiliates listed here. With that, this is H.V. Jagadish, Director of the Michigan Institute of Data Science, handing off to Dr. Anne Plant from the National Institute for Standards and Technology. Dr. Plant.
Thank you, John. And I would like to, I'm going to click the right thing here. Hopefully this will work. Yes, there we go. I'd like to thank you so much for the invitation. And so far I've had some really fun conversations. So I'm looking forward to this, to making this presentation. And I'd also like to say if anyone wants to stop me in the middle and ask a question, please do. But I look forward to questions at the end. I'm hoping that I leave plenty of time for that because already I've had some really good discussions and conversations. So I want to start by saying, giving you just a little background about NIST. And let me see if I can advance my slides here. There we go. So NIST is kind of a different place. We are the National Institute of Standards and Technology. We're part of the Department of Commerce. So we're not an academic laboratory, we're not an industrial laboratory, and we're a government laboratory in a government organization that isn't really a particularly science organization. But the important thing about NIST is that our mission is research in measurement science and advanced technologies. And our goal is to develop measurement science to advance technology and to advance standards, which advance technology. And so a lot of our mission is quite international in nature. So we represent the US as one of the international measurement laboratories, but we also represent the United States in standards committees and various other kinds of organizations that are critical for defining measurements and defining concordance and harmonization that impacts trade and it impacts, for example, regulated products and markets, and so it's this role of standards and harmonization, in addition to our research role, is really critical from a national point of view. And we consider this harmonization a word that I want to come back to during this talk a few times, which is this idea of comparability. So in other words, if some company or some company in some country makes some measurements, how does, say, the regulatory agencies in this country regulate that? There has to be a way that everyone has confidence about and can demonstrate the confidence that they have about their results. Now, of course, we work a lot with industry because that's kind of our primary function as part of the Department of Commerce. And so an example of this comparability that industry has to deal with every day is in a field that I work a lot with, which is the industry that creates cell-based therapy products. And one of the things about this industry is that it's still pretty nascent. There's a lot of discovery work still going on. And so when manufacturing of product starts, there are often a lot of changes in those manufacturing processes that occur. I think one of the ways that somebody has said is that it's like flying the plane while you're building it. And so what often happens in the process of creating a manufacturing pipeline is that manufacturing changes slightly. The manufacturing pipeline changes slightly, or maybe the starting materials or reagents change, the process changes. If you have a regulated product that has been awarded, say, an IND based on certain critical quality attributes, then if you've changed your manufacturing process, you need to demonstrate that you still have those same critical quality attributes. Those are measurements that have to be made, and they have to be comparable measurements, right? You want to make sure that you're measuring the same thing a year or 2 or 3 later under different conditions as you were measuring up at the beginning when you were doing most of the discovery work. So comparability means a lot to industry, and they understand that that good measurements and reliability of measurements is key to making a marketable product and a consistent product. This idea of comparability isn't so big in academia. A lot of academics don't really have that pressure. And so they have, a lot of people have different thoughts about the need for comparability. And they don't necessarily think as deeply as they might about the quality of their measurements. So, statements like, this process works for us, and our results are very reproducible, and our instruments are stable, and we know what we're doing. You hear a lot of stuff like that, and but that's not really the point, right? There are a lot of good academic researchers. Everybody does great work and cares a lot about the quality of their work, but comparability is different. Comparability says that that I can compare my results with your results, and you can compare your results with your results, 2 years from now. And that's not really so easy, as being able to compare results across different laboratories. And of course, that ability to compare results is part of what's required for sharing data. And sharing and combining data is just a huge interest. It gets bigger and bigger all the time, but we haven't really solved the problem about how to do it easily. So lots of good reasons for sharing data. I think everybody would agree that it would be great to be able to say combine and then reanalyze data sets and use multiple data sets to develop and test analytical methods and theoretical models, for example. A lot of good reasons. Discovering whether a particular study is generalizable to another model system, for example. And you can't really do that unless you can compare and combine the data from these multiple systems. And of course, as computer scientists, you all have an approach that's required for facilitating data sharing, right? It requires harmonization, and it requires transfer of data and security of data, and data storage and access, and all this has to be efficient, and vocabularies have to be harmonized. So, there's a whole lot of computer kinds of work that goes on. And of course, today, this idea of fair principles is really important. And so, there's more and more we're developing, I think, the infrastructure for the nuts and bolts of how to share data from a computational point of view. And of course, these are also federal priorities, national priorities. So there's some incentivization to do this, but this is kind of the nuts and bolts of how to share data and how to provide comparability. And it doesn't always work so well, right? So a lot of times, I'll go into a paper and that I've read and maybe interested in looking at the data, and the data are just like maybe spreadsheets or something. There's often not really a lot of information associated with data. Just numbers doesn't really do us a lot of good. And so, one might ask if this is a cultural problem. Do people not, are people not really that interested in sharing their data? Or maybe it seems like it's too hard. Are there real impediments to sharing data that could be reduced through data science and maybe better interaction between experimentalists and data science? So one of the kinds of data that we do a lot of collecting is image data, and particularly imaging live cells. So there's usually a lot of data that comes up, and we work very strongly. Our imaging program is very highly integrated with our information technology laboratory, particularly Peter Beji, whose name I've mentioned, I think, to at least one of the people I talked to today. And one of the things that they're very good at is developing ways of keeping provenance data for imaging. And this is a real challenge, of course, right? Because you've got a lot of pre-processing of an image, maybe flat field correction and maybe some background subtraction and maybe some filtering. And then you're going to do, algorithm development, and maybe you're going to try several different approaches, and you want to keep track of all of that and what the results of that were. And, and eventually you want to be able to go to every spatial location on those images and extract numerical data and analyze it. And so this is a very significant data challenge for making the information that goes into a result available to follow-on users. Really critical, because just the result itself doesn't provide you with enough information. You'd like to make sure that that the way that these data came about makes sense, and that if you do it a different way, does it change the result? So these are, science questions that have to be asked, and they cannot be asked just by looking at the final data. You have to have a whole bunch of information about provenance in order to make sense of this. But this, I chose this example to start with because this is a data science problem, right? And so it's probably not that unusual for anybody in the audience here to be thinking about this kind of data handling challenge. But in my world, which is trying to cope with biological systems, the provenance of an experiment, if you will, can become really complicated really fast. And it's one of the reasons I think that biological research is sometimes so hard to get a handle on and to really evaluate in terms of being able to share data and combine data, is that experimental metadata can be really complicated protocols, lots and lots of variables. If you're working with living systems like cells, like we do, then you've got time-dependent changes that are due to the vitality of and the natural changing that's occurring in the material that you're developing. And so, there are lots of things. So this schematic here was my kind of stream of consciousness after talking with a colleague a data scientist about Cordra, and so I tried to figure out how would I present this particular experiment that we're trying to do, which is we're constructing nucleic acid sequences that we're then using for genetic modification, for gene editing of a cell line. And each one of those gene editing events, transcriptions, is going to result in producing clonal cell lines, new cell lines that are modified. There may be several or many of them. Each one of those cell lines, each one of those clones is going to be expanded and tracked and measured in different ways, and things are going to happen to it. There are going to be lots of activities, and they're going to be analyzed. They're eventually going to be stored, and then thawed and used again. So a whole lot of things to keep track of in a very dynamic living system. And so, when I thought about, well, what are we really trying to do here? We could draw another schematic that looks a little simpler, which is dependent on a user interface. So the user is going to have input. This is the biological experimentalist who is going to be making these materials, performing these transfections on these cells, growing these cells up and doing various other activities with them. And what is that going to involve? So, we realize that we need some sort of sample designator and maybe a way of updating that designator as different activities happen. And then in addition to the user interface, of course, we want to be able to link this information to data that come out of instruments that might be on the network, or maybe come to us from other sources that have to be more manually input. But we could imagine perhaps parsing all of these and keeping them organized with a folder and file structure, right? So in a very simplistic, not very data science sophisticated way, this was sort of how we were thinking of keeping our information. And I wanna focus on this right-hand side of the screen, because I've had experiences with metadata and with trying to think about how to collect metadata, what kind of metadata should be collected, how it should be presented, and it's really hard to do. I mean, it's really hard to do. And one of the things that's really hard about protocol-based metadata, experimental metadata, particularly in a science that has lots of steps and lots of manual steps, is that it cannot be a burden to the experimentalist. If you've given the experimentalist that already has so many operations going on every day in the laboratory and a lot of manual things and keeping track of stuff, if you give that experimentalist one more task to do, like go back and fill in a metadata sheet, that's not going to happen. It's just not going to happen. And we're talking metadata here like that the experimentalist is going to be creating in the course of the experiment. Metadata that you can get off of an instrument, of course, in a header file, that's relatively easy. But the metadata that the experimentalist generates, much more difficult, right? And so my approach was to say, listen, if we're going to be able to capture all of the things that are going on in this complex scheme of activities, it's got to fit into this person's workflow. It's got to be part of their workflow. It's got to be easy to use and intuitive. It can't be something that's a form that you fill in afterwards, because you're going to forget. A couple of days go by, it's too burdensome, and you can't be just transcribing information from another page into your metadata sheet. And so I also sort of bit the bullet and I said, we're not going to design maybe it's silly to even think anymore about, at least from my perspective, to design metadata for the general case. Instead, we really are just going to focus on the specific activity, and we're going to make it something that the experimentalist or somebody without programming knowledge can easily modify and update as needed. And of course, it would be great if it can be transformed eventually into XML or some other kind of useful database format. And so what we turn to And, you know, I say this just a little embarrassed, but it actually was pretty successful. And there may be a lesson here, is that we turned to Excel, using Microsoft Excel as the user interface. It allowed us to capture when we did these transfections, these gene editing events, when we selected clones, how to designate them, and then for each clone, all of these actions could be recorded, and through Excel, we could capture not just the events but the dates as well, and then this information could be linked to other information, other data about these samples, and we did this by by watching how the experimentalists did their workflow. I mean, we really let this person start in on the process, and then looked over his shoulder and said, how does he keep his data straight, and how do we make that easier for him to do and make sure we capture all the information we have to? And what we did was we started with a calendar format. Now, we did use a professional Excel programmer to do things like set up this functionality of a calendar, and a few other bits of functionality. But in a context where we were able to go in without any programming knowledge and make changes and update the protocol collection, the information of the protocol information collection is pretty straightforward and easy to do. And so what the user can do, the experimentalist can do, is for any day of the week, he can see what he needs to do that day. He can make a list of all the cells that he's going to be doing some sort of activity on. He can go back and look at what happened last week and think about what's going to happen the next week. And so it really helped the experimentalist in terms of keeping his workflow well organized. So it had that benefit. And then it was also really easy through Excel to create pull-down lists, so that as the experimentalist is putting data in, they don't have to think about what's the name of that cell line. They can just go select a cell line. And some of these cell line names get, because they're evolving over time as new activities are happening to these cells, the names are similar to one another. It could be easy to get confused, and it's much less easy to get confused and make a mistake if you can just select something. These cell names actually were updated automatically through entries that happened at the calendar level. And so this list of cell names just was updated automatically and could be pulled from. We also could create entries for other things like the activities that would be done on these cell lines every day. And again, this was this pull-down menu that is really easy to update. And this is just an example of the kind of page that you can set up in Excel with pull-down menus of information that you can designate. These are limits on the data that you can choose to put into those into the forms on the other pages, and it's very easy for an inexperienced user to go in and add to these lists or change these specifications as the experiment might change, as you realize there's another variable that you should be keeping track of, or as you realize you need to change to a new medium, or you need to change your protocol a little bit. This kind of functionality of these pull-down lists is something that is embedded into the toolbar in Excel. It's very, very simple for the user to, the uninitiated user, the not very well educated user to learn how to use, and it makes it very simple. And so having that adaptability was really, really helpful. This kind of thing also gets used in at the beginning of the process of creating these cell lines. That transfection gene editing activity that gets done is one could make very easily a template to collect the metadata that's associated with the cell line that you're creating, with the transfection that you're creating. And again, using pull-down lists to select designations for the materials that you're using, the nucleic acid sequences even that you're using in the process of this gene editing protocol. And to add a little more value, we embedded in calculations into this form so that as the experimentalist is trying to figure out, let's see, how many cells do I have and how much material do I need and what's my final dilutions, this can all get calculated on the sheet. And so there's actually benefit to the experimentalist, and it's a little more incentivized, I guess. And again, something very simple to do within Excel and easy for a non-programming person to develop. One of the other benefits of this Excel approach is that all of the data that's collected from the calendar can go into a data sheet, a list, basically a data list. With all the information you need, including information about the user and the computer that generated these data. And through, by combining Excel and Access, another Microsoft product, one can turn this into an XML. And so, it makes it easy then to transition all of this information into a database. So, this has become a tool, as simplistic as it is, but I think that it might be a good example of how getting the experimentalist involved in thinking about how they can easily collect metadata and working with a data expert to make it as easy as possible. I mean, I think we have to think about how do we lower the bar, as some of us talked about earlier today, how do we lower the bar to create tools to make it easier for people to do the things they want to do in science, do them as reliably, responsibly, reproducibly, if you will, as they can, and make as few mistakes, collect as much information that then can be added to the data stream, so that it's easy for others to follow all the details that were actually done, and to then do query about how the cell lines that you end up with might have been influenced by some of the specific aspects of the protocols, how many days in culture or what medium was used, for example, all those variables that you might have otherwise lost. So, I would contend that metadata is essential for sharing data, for assuring comparability of data, and for establishing confidence in data. And this would be, if somebody were asking me what my opinion is, this is my opinion, is that to be maximally useful, that data, it's not just about the data itself, but about the quality of the data and the qualification of the data. That's what you really need as a basis for comparability and confidence in data that will make it reasonable to share and make it easy to share. There have to be highly granular, there has to be a way of capturing highly granular experimental and protocol details. Or otherwise you have no way of comparing those differences that might in fact be differences that mean that you're doing a different experiment than I'm doing, because we aren't actually using the same protocol, even though we think we are. The details are different, and the details are variables that make a difference in the outcome. One of the other things that one could work into metadata, which I think would be excellent, is ways of reporting uncertainty or other indications of quality. And I'll talk a little bit more about uncertainties. Ways of capturing benchmarking data from reference materials, for example. And of course, qualification of software. I mean, all of this are examples of metadata, but they might be examples of metadata that often aren't captured. But this does, I think, define the quality of the data. And if one's going to care about quality of data, which of course is going to determine how useful those data are to share, it's really important to have that documentation of the quality of the data and that thought about the quality of the data. So, NIST being a measurement laboratory, we think a lot about tools that that help to add to quality of the data, what we might call tools that help to achieve comparability and confidence in the data. And so those tools might include reference materials, like, this is a reference material for benchmarking microscopes. It might be assays for analyzing cell lines and whether or not the cell lines are coming from the species that you think that you're using. It might be sending samples, identical samples, out to different laboratories to, for example, do whole genome sequencing, and then come back and say, well, how did your whole genome sequence compare to mine? And were the errors in the sequencing, or were the differences in the bioinformatics tools that were used, and allow evaluation then of the quality of those tools. Really important, and this was an example of an international level study involving toxicity of nanoparticles. So, it has implications, is using a tool like an Ishikawa diagram to systematically look at your sources of uncertainty in your study, and then figuring out where your most important sources of a certain uncertainty are, maybe by doing a design analysis, and then using that variable space and controlling that variable space to take data from different laboratories and make them comparable, so that you can actually compare them to one another and say, oh yeah, we are measuring the same thing, and that material is more toxic than another material for sure, not just an opinion. So, so these are ways that one can think about perhaps adding these kinds of data into metadata that really enhance the quality of the data that come out at the end. So I want to say something about reproducibility, since that's on the tip of everybody's tongue right now, and the fact that this reproducibility crisis, as it is sometimes called, and I'm not sure that I really agree with that, really came out initially through research done at pharmaceutical companies that were trying to reproduce published studies in biomedical research. And the studies that they were often, well, in both cases, the studies that they were trying to replicate were studies that were published in very high-end journals. They were breakthrough discoveries and observations. And then it turned out that a very small percentage of those studies were reproducible in the hands of the pharmaceutical companies that were trying to further that research. And so, this set up a big consternation and got a lot of press in many different formats, and a lot of concern from the federal level about, are we failing? Is our research enterprise failing because of irreproducibility and the publication of irreproducible results? I just want to point out that that this is not an issue just for the biomedical community, but rather all communities have a certain amount of I guess, concern about the veracity or the care with which data might be published. So this is work that comes out of NIST from our thermophysics laboratory, where they have agreements with a couple of journals where they will look at a paper before it gets published and run it through a semi-automated checking method to determine whether there are errors or surprising results that aren't theoretically possible in the paper before it actually gets published. And 30% of the time they find that there are problems with the papers that could be dealt with before publication. And so, one of the recent conversations in this area was taken on by the National Academies. It came out with this report just a few months ago. And my colleague Bob Hanish, some of you may know him, and I contributed to some extent to this report. And we were pleased with the outcome, because one of the things that this report points out is that while irreproducibility the inability to independently confirm a computational result may indicate a lack of rigor, it's also possible that these inconsistencies are actually important components of the science, that they give us more information. Maybe it's a new discovery. Maybe it's surprising that even though you thought we did the same thing, you didn't get the same result. And why is that? When you ask the why is that, that's when you perhaps learn something that's important to learn about the scientific system that you're studying. And I think that's a really important and useful way of looking at this problem. And so, I've been, hoping that that more and more efforts would be made to kind of reframe what has been called the reproducibility crisis, and get away from that idea of thinking that reproducibility is the be all and end all. Reproducibility is not a guarantee of accuracy or of truth. A failure to reproduce a study result may be because the experimental conditions are different, right? And now you're learning something about the experimental conditions. It's not that you have done something wrong. And also, many observations are one-time events. You're not going to reproduce a hurricane to be exactly the same thing, or a volcano eruption. So, so there has to be some option here to just thinking about reproducibility as being the most important thing. And I think the most important thing that we really care about is having confidence in measurement, right? And so that's what we should be aiming for. How do we establish confidence in measurements? So confidence is a component, or arises from a number of metrology related components, and reproducibility is just one element of that, of those components. So this is a list that is often used to describe an assay, for example, and what are the components of an assay that are important to establish one's confidence in the results of that assay. And you can see that reproducibility is just one of those, right? Accuracy and precision are completely different from reproducibility. They have different criteria. The ability to get the right answer, even if assay parameters change, to have a reasonable, useful dynamic range of response function, to not be too sensitive to matrix effects. These are the things that allow you to qualify an assay. And again, reproducibility is one component, but not the only important thing. And so, to kind of build on this, we really wanted to think about how, if we were to look at the scientific enterprise at large, where many different kinds of research are being done, it's certainly not all biomedical. Every field of research, computation and theory, materials science, whatever, every kind of research that gets done really has an onus to look at the same qualifications, I think, or to have the same issues and responsibility to look at uncertainties. Uncertainty is really what you want to look at as an indicator of the confidence that you have, or how you establish confidence in your research effort. There are always sources of uncertainty. Those are variables that you may or may not have control over. If you identify variables that you have control over, then you can mitigate those variables with some of those tools that I showed earlier, benchmarking materials, or design of experiment. But if you have variables that you don't even know exist, and you don't know what their effect might be, then the best thing you can do is at least understand and acknowledge that you have sources of uncertainty that you have not controlled. And so that's really important. I think that one can look at that concept of what sources of uncertainty go into your research effort, regardless of what research you're doing, at every stage of a research endeavor. And, with respect to experimental data, that's a component, but there are a lot of non-experimental issues that go into all of our research efforts. And so, if you want to look across the range of all the activities that go into your particular research program, and identify the sources of uncertainty, you can start with planning. What assumptions are you making? What previous paper was published that you're basing your experimental protocols on? Do you have complete confidence in those protocols? Do you have confidence in the assumptions that were made in that other paper? Did they support their assumptions well? What is the generalizability of the research that you're doing? Are you going to say, you saw X, Y, and Z, full stop, or are you going to say, well, you saw X, Y, and Z in this particular experimental system? Reagents, equipment response, these are all relevant to an awful lot of different kinds of experiments and different kinds of studies. Quality of the experimental data. There are ways of minimizing bias in data reduction and interpretation of results. And I guess that this is probably the most important thing for data science, right? What is the basis of a statistical test? Are you using the right statistical test? Why did you choose that statistical test? What was the basis? And is it appropriate for the data that you have? How are algorithms being evaluated? How do your data compare? How do your results compare with previous data? Have you identified uncontrolled sources of uncertainty? Have you considered alternative interpretations in how those results, what those results might mean? It might be really easy to interpret your results in a way that someone else interpreted their results, but are there other ways of interpreting the results that would still be consistent with the data? And of course, the thing that I really want to focus on for this talk is the idea of reporting and disseminating all of the information that goes, or as much of the information as you possibly can, that goes into this research study. As you all try to do here, documenting the data and the code, more difficult perhaps to document protocols and process for an elaborate experiment. But all of this is part of metadata, and metadata has to be a really important part of reporting and disseminating if we're going to achieve confidence in research and facilitate data sharing. So with that, I'm going to conclude my talk with these points that hopefully I've made and will stick with you and make sense. Is that reproducibility isn't always sufficient or necessary as a criteria for a research result. That what is really the most important thing is establishing, with documentation, establishing confidence in the data. That data can be established, that confidence can be established by really thoughtful consideration of sources of uncertainty, and then thinking about mitigating those sources of uncertainty with some of those tools of metrology. I do think that perhaps the single most important aspect of having confidence in data is good reporting, and good reporting of not just the data, but the metadata. Just the data it's not really going to help anybody. You have to understand the context of it, and you have to understand the details of how that data came to be. I would put this out there for both experimentalists and data scientists that metadata capture, we have to figure out how to facilitate metadata capture with tools that make the process inherent to the experimentalist's workflow so that it is not burdensome. And we also have to think about how do we bring experimentalists into the data handling process, into the data collection process, because they have to have control over grabbing that metadata from their experimental workflow. They have to figure out how it works for them so that it does work. And if all this stuff works, then I think we're able to really establish confidence in research and be able to share our data a lot more effectively. And so with that, I will thank you and take whatever questions you have.
Well, thank you very much, Dr. Plant, for an inspiring talk. I'd like to request people who have questions to post them either at the Q&A button or the chat button that you see at the bottom of your screen. And while you're doing that, I'd like to start by asking, in terms of how one gets scientists to adopt things. You said a number of things that resonated strongly with me in terms of things that we've been doing. So as a computer scientist, I was working with some material scientists and as experimenters trying to get them to adopt a tool and record their data in a way that was shareable and reusable. There's a question: what's the workflow and what's the return they get immediately for doing this? There's a long-term return in terms of a few years later coming back even to their own work sometimes. But I think that at least in my case, I felt that the solutions were specific to that particular type of work and type of data. And it would be nice if we had some broader, bigger theory or principles for how to do this, or how to take what you've done for a community and say, here, I can now press this button and turn the crank, and I can do this for 6 other communities, making appropriate small changes. How far do you think we are from that? Are we, in many fields, there's always, it's not unusual to do things first in simple examples and then sort of understanding the general principle. But how far do you feel we are on that journey?
Yeah, no, I hope that that we're farther than I think. I mean, I watch, I watch metadata acquisition systems that other people have used. I work with a small community of people who are all interested in LIMS, and they're mostly data scientists, although some experimentalists. But I look at the systems that they're using for collecting metadata, and I have to say it looks really cumbersome, right? And again, as you say, it's really not that easy to convince somebody that 5 years from now they're going to be glad they took the extra 30 minutes every day to fight with some metadata acquisition system. And so this is why I think that there's a real onus on the experimentalist to think about what their data flow is, what their workflow is, and how they would design something that was helpful to them. Because once the experimentalist who's actually going to use this, and if we thought of it that way, this isn't a data science issue so much as it's an experimentalist issue. If we put it on the experimentalist and said, think about what would be helpful to you. Like if you had this and this and this, would this help you on your day-to-day getting your work done? I think that's the thought process we have to go through. And that's the experimentalist. The experimentalist has to start that process. But once the experimentalist is, and this is why I liked the Excel thing, is because we could just play with Excel really easily. It didn't have to work every time. It didn't have to be perfect. We could see that there was functionality that we'd like in there, but we weren't experienced enough with Excel programming to do it ourselves. But once you had the outline of how you wanted to collect data and what data you wanted to collect, it's really easy then to go to a data scientist or a programmer and say, could you codify this for me? And particularly in something like Excel, where then part of it is stuff that, is really easy for you as the experimentalist to change and modify. So I think it's, I think it takes, again, teamwork, like all the hard things. But it really has to start with experimentalists. And I'm not really sure how one makes that happen. I think one of the things that I noticed working with our biologist who was doing this work is that it's really hard sometimes for some people, although I think younger people don't have a problem with this so much anymore, to really think about machine readable data and the fact that, yeah, you want to make notes and you want to have a space to make notes because there are some things that, are going to be surprising and you have to capture that information. But most everything you want to think about in terms of a variable. Is this a variable? And therefore, can I capture it in a machine readable way? Maybe that's the hardest thing for some experimentalists to get their heads around. But I think it's got to start with the experimentalist. I don't think it can start with the data scientist. But then once there are enough examples of this, probably, brilliant data scientists will figure out how to make this a more turnkey kind of approach.
Thank you. There's a question in the chat from Abby Jacobs.
Oh, I see. I've been thinking about similar problems, but more in the social sciences and computational systems. A challenge in this context and yours is about making assumptions explicit to better assess validity, reproducibility, etc. Can you talk a little bit about how you see that fitting in with designing systems for collecting metadata? Yeah, that's a very good question. I mean, that idea about thinking about assumptions. Again, it could be a line item in a metadata sheet that, and maybe maybe one could reduce those assumptions to a set, that could be part of a pull down menu so that the experimentalist didn't have to write a thesis in order to complete a metadata sheet. But yeah, that doesn't fit neatly in what I showed you. Yeah, sorry, but I think you're right. I think that it's really important to assess assumptions and make that explicit.
Sometimes people may not even know. Sometimes people may not even know what their assumptions are.
Absolutely. Yeah.
That's very hard to surface.
Well, this is I guess it was Jing that I was talking to about this. There's been so much consternation about this reproducibility thing, and I've watched NIH and worked a little bit with NIH as they've tried to develop checklists, and then a bunch of journals have developed checklists, and I think checklists are just the wrong idea. I don't think you want a person who's preparing a paper to say, yes, I did this. Yes, I used statistics. Yes, I, blah, blah, blah. I think you want a person to think instead about what was the study I did, right? And at every step of this study, what were things that could have been sources of uncertainty, like making assumptions, and what assumptions did I make? You're never going to find that on a checklist. It's got to be a thought process that the experimentalist goes through, I think. Should I go on to the next question?
Please go ahead.
Zachary asks, do you think that by exposing all the data from so many various sources, we risk the misuse of data by people taking machine readable formats and neglecting to pay attention to all these meta factors? Gosh, I'm not sure that I understand, Zach, what you're talking about, but I think what you're saying is that if you just have numerical data and you don't have the context around it about how it was collected or how it was processed or what some of the, whether or not it was benchmarked in any way, that, yeah, it could be perhaps misinterpreted because there's not enough metadata associated with it. Yeah, I don't know if that's what you mean, but I could see that that could be a problem.
So the last question we have is actually from in the Q&A button. That's from Jing.
From Jing. Your list at the end with reproducibility being one component for data reliability and confidence is very refreshing. Okay, good. There are definitely pushes for various definitions and approaches for research reproducibility or the comparability of results. For example, the NAS definition of reproducibility is focused on whether one can rerun somebody else's workflow. How can we get on the same page about what's essential when so many discussions are happening? Well, you know, Jing, one thing I would say is that I'm glad that there are discussions happening, and the discussions are getting more and more sophisticated. You know, you're not talking about checklists, right? You're talking about, like, how do you think through these things? So I think that's really good news. The fact that that we haven't, as a community, and it's a very wide community, decided on a definition for terms. I mean, I see that as less of a problem than being clear about, you know, defining what do we mean by this. So, you know, I think that's more important. Now, you know, I throw out that list that, you know, after a lot of thought, we put together and decided to publish it. But that's just fodder, you know, that, I mean, that's just, to get the conversation going, as far as I'm concerned. I don't think that's a list that anybody should think of as in stone. You know, it's a list from which we might be able to develop a better list, or think about what is important in a particular research space. You know, I think that it's going to require a lot more discussions, but at least the discussions now are about reliability, and they're about confidence, they're about uncertainties. They're not about the sociological pressure on researchers to publish quickly and publish sloppily. So I think that's a benefit.
Yeah, with that brings us to the end of the hour. Just been really a fascinating hour. Thank you so much for
Thank you so much.
Opening our minds and helping us move forward on as we all think about reproducibility and related topics. I appreciate
it was my pleasure. It's quite a delight. Thank you so much.
Thank you, and see everybody next week. Bye-bye.